Okay. Hello, everyone. So welcome to the webinar hosted by MetaCell today about extending the Neuroglancer ecosystem. So we've got a really great set of speakers today, and we're looking forward to hearing what they have to say. You're probably familiar with Neuroglancer, but just briefly, it's a browser-based viewer for volumetric data, and it's capable of displaying raw images, meshes, and skeletons while all staying highly performant despite being in the browser. It was developed at Google, and there's been a number of contributions to the community as well over the years, and it's currently maintained by Jeremy Maton-Shepherd. So thanks, Jeremy. What we'll be talking about today, though, is extending Neuroglancer. So things building on top of Neuroglancer, or sometimes changing Neuroglancer itself, and then the kind of challenges you run into when you start to scale up to these large images in the browser, particularly when you want to collaborate on them, whether that's with the community abroad or internally. So that's what we'll be going through today. It'll be a 90-minute webinar. So if you can stay to the end, great, but it'll be recorded. So no problem if you can't, you can watch it afterwards. We'll do a quick round of introductions to begin with. And then each speaker will have 10 minutes to present their topic. So that includes their topic plus some time for QA if it fits. And then it'll move on to the next speaker. With that all going to plan, I believe it's about 10 minutes at the end or so for a final wrap up. So any questions we didn't get to the speakers or any questions that maybe go around the panel in general, and then we'll wrap up there. So with that, let's start the introductions. I am conscious that we have 7 speakers, so we'll do the introductions quite brief for now. You can always expand during your talk. So we'll just do who you are, what your affiliation is, what you'll be talking about, and if you want to, where you're calling in from. So I'll go first. I'm Sean Martin. I'm calling in from Zurich. I do software engineering at MetaCell, and I've been directly involved with some of the changes we've been doing with Neuroglancer, along with the Allen Institute and CZI. And today I'll just be helping to facilitate this session. So Forrest Coleman, if you could please introduce yourself.
Yeah, my name is Forrest Coleman. I am an Associate Director of Data and Technology at the Allen Institute and a co-lead of the EM Connectomics project. I'm dialing in from Seattle, Washington today.
Thanks, Forrest. Ari Matsuo.
Hi, everyone. My name is Ari. I am a research scientist and data evangelist for Flywire at Princeton Neuroscience Institute, currently in New York City.
Thanks, Ari. Lars Yesterby.
Hi, I'm Lars Yesterby. I'm a technical staff at MIT Lincoln Laboratory. I'm leading the team that put together NeuroTrail, which is a technology for neuron tracing and active learning, and calling in from the Boston area.
Great, thanks, Lars. Yael Bastre, please.
Hi, yeah, I'm a fellow at University College London, but this project is really mostly based in Mass General Hospital in Boston. And I'm going to talk about using Neuroglancer with human neuroimaging data and coordinate spaces.
Perfect, thank you. And Stuart Berg.
Hi, yeah, I'm Stuart Berg. I'm project scientist for the FlyEM project at Janelia, and I'm calling in from Virginia.
Perfect. And David Ackerman.
Hi, I'm David Ackerman. I'm also at Janelia, and I'm a software engineer working on the CellMap project team, where we segment organelles from large-scale volumetric EM datasets.
Okay, and Matteo Cantarelli.
Hello, everyone. Thanks, Sean. My name is Matteo Cantarelli. I'm an engineer by background, CTO at MetaCell, and one of the co-founders. Today, I will be talking about Neuroglass, which is something we've been building in the past year on top of Neuroglancer, and curious to hear what everyone thinks about it. Thanks, Sean.
All right. Thank you so much.
I'm in Italy, in Cagliari.
Good, thanks, Matteo. Okay, so we'll keep that same order for the speakers. So what we're going to do now is everyone's going to run through their 10-minute talks. So the talk will be, as I said, 10 minutes in total for both talk plus the QA. So anytime you have at the end of that 10 minutes, essentially, it can be used for any QA from the audience or from the speakers, or failing that from me. But we do really encourage questions from the audience. So please just put whatever you're thinking in the chat, whether it's questions or thoughts, and you can send them to everyone because then you can see what other people are thinking as well. And then we'll try and get to them when people come along, whether part of the questions or when we get to the end, we should have a little block then to ask any of those that get left over. So with that in mind, I am going to pass it over to Forrest to kick things off.
All right. Thanks, everyone. So today I'm going to talk about two different kinds of collaborations on Neuroglancer. The first will be the collaboration between the brain science and Sebastian Sung's lab at Princeton on CAVE, the connectome annotation versioning engine, of course, with help from Jeremy at Connectomics at Google. And then the second part, we'll talk about some non-electron microscopy use cases and features that we've developed in collaboration with Neurodynamics and MetaCell, along again with collaboration with Jeremy. So first, CAVE. CAVE is a sort of infrastructure for doing large-scale editing of electron microscopy data sets that was developed in collaboration between the Sung Lab and Brain Science. The team here is shown on the left, and we've had a number of contributions from outside others organizations as well. This is the infrastructure that backs many of the large-scale electron microscopy reconstruction projects, maybe most notably Flywire and Microns, but also HO1, Fancy, Bank, if you know what those things are. There are now over 20 data sets from more than 10 institutions which are being utilizing this infrastructure. Briefly, why do we need this? In doing large-scale reconstructions, you get sort of dense segmentations out of these automated methods, but there are many mistakes. You have a neuron, say on the left, you need to add and remove portions of that neuron to get a proofread neuron. Simultaneously, people are making observations about the data set, either with machine algorithms or by humans, annotating things like synapses and mitochondria and cells and cell types. And all of this data needs to be put into a centralized infrastructure so it can be queried by scientists, and that needs to happen in real time as people are continuously adding proofreading and annotations to this data set spread out across the globe. And so CAVE is a set of web services that enable this kind of use case. So what does this look like in Neuroglancer? Here's a brief demo that shows proofreading of a neuron. So this red neuron here, the yellow dots represent output synapses that are detected in the segmentation. And here you can see some output synapses that are coming off a dendrite, which is not correct. There's an error, a merge error. And so here the user is able to put down some red and blue points that illustrate where the two objects are, and then use that to submit to the server, which will dynamically calculate where that segmentation should be split in order to segment the purple and magenta objects here. You can see that the synapses were immediately associated with the purple objects, so they could be removed from the view. So how do we pull this off? There basically are two strategies. One involved adding a custom data source to Neuroglancer through the server called the PyChunk Graph server. This is a unique data source that has some novel APIs that allow for this, such as merge and multicut that I showed you, as well as get leaves and get root that are involved in being able to quickly load a dynamic segmentation. Other features we've implemented by creating servers that mock static formats, namely the pre-computed format. So we have servers that produce skeletons, segment properties, and annotations that are served to Neuroglancer, and the server essentially mocks the format to allow that data to be served. I'm going to focus now on kind of what we had to do to Neuroglancer, or what are some of the features that we've added. The first point I'll make is that this has been going on between the Sung Lab and CAVE for a long time. From 2018 to 2022, there was kind of the Sung Lab master branch, which unfortunately got forked from mainline Google in a very painful process. You can see it's currently 237 commits ahead and 928 behind. Since then, we've abandoned that fork, and we've been trying very hard to stay up to date with mainline Google in this Spelunker branch, which is currently 29 ahead and 1 behind of Google, which is deployed at spelunker.caveexplorer.org. And we're trying to merge things in there that we're pretty confident are going to get into mainline in the end. Some examples of features that we have there that aren't yet merged is a custom color picker, which is, I'm noting all the PRs that are below these features here, as well as the ability to iterate through annotations and toggle tags on and off, which is this PR. And then finally, a build time option for global hotkeys. So for when we're doing proofreading, we want to make a user interface that allows the most common operations to be very easily accessible via keys. And Neuroglancer is a little opinionated about the way in which hotkeys work. This allows our kind of compromise position to allow for a build time option that allows for global hotkeys to be programmed to certain elements of the UI. A couple of things that so I should mention, Chris Jordan is the person that's doing most of our Neuroglancer development in CAVE. Here are a couple features that he's been working on lately. One is a focus mode for mesh loading. Currently, this is implemented for graphene that allows the user to sort of focus only on a portion of the mesh, but perhaps this should be extended more globally. And the second is allowing for skeleton shaders to have physical line widths that are modified by vertex properties, maybe most trivially to use the radius of the skeleton so that you can see what the skeleton actually looks like, but also to make it so that it maintains a single pixel line width as you zoom out. And then a less UI focused, but nonetheless important PR that Chris has worked on is rate limiting the URL updates for large states so that your Chrome history is not bogged down with a bazillion very large states. We've seen Chrome really struggles in that context. So I'm going to pivot now to the second half of non-EM use cases. This has been a collaboration. Brain Science uses Neuroglancer, or the Allen Institute uses Neuroglancer to visualize very large images, both EM but also non-EM images. And we've been partnering with MetaCell to develop some new features, and Google for developing features that are aimed for this. So the first is about multi-channel. So we've added in features that allow for these min-max sliders to the invert features to be more performant and easy to use. So there's this idea now of shader controls that are built into Neuroglancer where you can group all the shader controls from different layers into a single palette. And you can see there are now min-max sliders. And there's a PR open that simplifies setting up these multi-channel, say, OMI-ZARs based upon the metadata, putting different channels in different layers and automatically setting the colors and min-max values. Another feature we've worked on is adding screenshots to Neuroglancer. So this involved both making this field of view parameter editable in the UI, where you can set the field of view or the resolution in order to get exactly what you want. And then the screenshot UI, which shows the users exactly what data at what resolution is going to be pulled and allows them to pick upsampling to get, quote, high resolution screenshots. And you can name your screenshot and then take the screenshot. And what you get is just what you'd like to paste into a presentation, namely no UI controls, just the sort of rendered portions of the panel. Another important thing for volumetric light microscopy was the lack of true volume rendering. And so this was a feature that was added in collaboration with MetaCell. So here's a many terabytes of scale light microscopy image taken for the exospin microscope. And you can see we're doing volume rendering here in the browser of this very large data set. And as you zoom in, it's loading progressively higher resolution data. And you can navigate around just by right-clicking, and it will use the sort of path tracing to find the point of highest signal intensity to help you jump. That is particularly relevant for the next feature that we added, which was polyline annotation tool. So this is adding a new annotation type to local annotations, where you can now have a series of lines rather than just an individual lines. And so the user can either in 2D or 3D lay down, trace this fluorescent volume in a local annotation. This is very much inspired, I think, by work that NeuroTrail and others have done in this area. And then finally, recently, there's a draft PR open for having annotation property schemas and editable annotations within Neuroglancer so that you can have your annotations also associated with numbers or colors or enums and so on and so forth. And so we're looking forward to that being completed very, very shortly. So with that, I'd like to thank everyone that's contributed to this. Most importantly, Chris, who's done a lot of the work that I highlighted, and Sean from MetaCell, as well as, of course, Jeremy, who has been an essential advisor and reviewer on all of these features. So thanks.
Great. Thanks so much, Forrest. We don't have any questions from the audience just yet, but feel free to post them in the meantime. That was fantastic, though. So in the meantime, I'll ask something myself. What I was wondering was with Cave. So Cave is really, really interesting. I've looked into it outside of this as well. But for people who would consider using Cave, is there kind of a time, when they should consider using it? Like, is there kind of a tipping point at a certain scale or a data size where maybe they should start considering that as a backend for their annotations versus when maybe something that's, not as powerful, but a little bit simpler might be appropriate?
I think that Cave is aligned around the use case of having very large data sets being edited by many, many people. And so if you're doing a project that requires many humans to collaborate on a very large segmentation that is the segmentation is likely to change, and you anticipate the annotations are also likely to be changed, and you want to, have many groups and people contribute to both of those things. That's the use case that Cave is designed around. If you're just a single user who wants to do their own annotations, and collaboration is not really important, or your data set size is not a big volumetric segmentation, then I don't think Cave is, the complexity of setting it up and the cost of doing so is probably not worth the squeeze.
Makes sense, yeah. We have a question from Wyatt, which is efficient splitting of supervoxels on the roadmap for Cave? And if so, how far out?
It is on the roadmap for Cave. Basically the issue is we're working on trying to have have a dynamic kind of the supervoxel watershed layer needs to have a dynamic serverless backend. And so we're targeting the this called OCDBT, which is a feature that Jeremy has developed at Google to be the backend for that. We have a lot of the low-level stuff worked out, and now we're hoping in the next 6 to 12 months, we're going to have our first implementation that will implement one algorithm for doing supervoxel splitting. We're going to do something heuristic at first, but with the idea that there might be multiple algorithms that would slot in there, like maybe you have a custom neural net that you're going to use to rerun segmentation in those cases.
Great, thanks, Forrest. We've got some other questions, but we might get to those later on in the panel. For now, we'll move on to Ari, but thank you again.
All right. Can you hear me okay?
Perfect.
Okay, my name is Ari Matzlia, and I'm gonna speak about the Flywire Codex. Myself, my background is theoretical computer science, graph theory specifically. And after about 10 years in tech, I came back to academia in 21 to work on the Flywire project. For those that are not familiar, Flywire is a consortium of many labs and hundreds of scientists worldwide, including citizen scientists. It is led by the Murthy and Song Labs at the Princeton Neuroscience Institute, and the mission of Flywire is to map out completely the drosophila nervous system. This is on the bottom. You can see we had a big publication of the full brain data set last October. It made a lot of waves in the news, and this is a screenshot on the right-hand side of the reconstruction. This is 50 largest cells in the fruit fly brain. The topic of this presentation, the Codex, is a data explorer for connectomes. It is designed for the stage where the connectome is fully reconstructed or close to fully reconstructed, and it is designed also for anywhere from scientists as well as high school students through our program that's called Flywire Academy. The Codex is fully web-based. It doesn't require any downloading of data or code. And so far, it has logged about a little bit more than 50,000 registered users. And on typical workday, it will serve about 1,000 search queries per hour with a latency of half a second on average. And so I want to talk a little bit about the Codex itself, how it utilizes Neuroglancer. There's many different integration points. And I will also briefly mention kind of the design principles of Codex and compare it to Neuroglancer. Here is the top kind of like the popular apps in Codex, just taking screenshots. It's here just on the left top, you will see the landing page for Codex, where you can select the data set and different analysis applications. Then on the right-hand side on the top, you can see the most popular use case for Codex, which is searching up cells by annotations. And so this is how the search results will look like, with all the different links going out and branching out to other pages. And on the bottom, you see a detail page for a particular cell. In essence, every single neuron that's in Codex has a homepage where you can see all the different information that's available for the cell with links outside to other tools that have information about the cell or cell type. And you can also see the Neuroglancer embedded in this view here. So to briefly mention how Codex is built and compared to Neuroglancer. So as we all know and enjoy using Neuroglancer, it's a very generalizable and flexible design where you basically get a static page from a web service, and then all the logic and all the kind of brains of the application happen in the browser. It will load the images or the meshes in the most optimal way directly from your browser. Then on the other extreme of the kind of design spectrum, we have the full-fledged web services that are backed by database, like in K, for instance, or something like Neo4j, where a user request will initiate a lookup into a database, and then a view will be built and sent back to the user, or data will be compiled and sent back as for programmatic exploration. And the Flower Codex is somewhere in between. It is a code-only service. What I mean by that is that it's not tied to any centralized database. All the metadata that is used inside the codex is loaded on startup. And then all the user requests are served in memory. So all the computation happens in memory, whether it's a search by metadata or it's graph exploration algorithms. All of that happens in memory, and then it will typically either compile a HTML page to send back to the user or a Neuroglancer view that corresponds to the query and will send the kind of compact configuration back to the user to render in the browser. So you can think of the pages that utilize Neuroglancer and Codex as a kind of layer that maps user queries to configurations and kind of avoids this complexity of sending the entire batch of metadata to the browser and then sorting through the which meshes to load or which images to load in the browser. And I want to highlight a few examples here, going from the bottom left, the simplest example where you just punch in Codex some cells and you can visualize them. You can tweak the configuration. You can basically color by annotations or some other attributes. Then in the center bottom, you can see now the query is a little bit more involved. I'm asking here for pathways from one cell, which is a photoreceptor, to another cell, which is a descending cell. And then Codex will find all the cells on the pathways, shortest path, kind of intermediate cells. And it will send back a configuration for Neuroglancer to load them and to mark them, the connection points and so on. Then on the bottom right, you can see another use case for Neuroglancer and Codex here. I'm looking for motifs. In this case, I'm looking for 3 neurons that are strongly connected so that every pair is both upstream and downstream partner of each other. And then every single instance of such a motif in the flywire connectome can be visualized in Neuroglancer here in the embedded view, again, with showing the synapses where the cells connect and so on. On the top level is the busiest view. It's all the medulla intrinsic cells in the optic lobe of the fruit fly. And in this case, I'm using the metadata in Codex to color them by their distance from the center. From the center of the retina. And in summary, so the Codex is a browser-based tool that utilizes Neuroglancer in many different applications. It is backed by in-memory search and exploration of the data set, which allows us to scale it quickly up and down based on demand. There is no bottleneck of a centralized database. That's a design principle that allows us to basically utilize the resources to the maximum. And you are all welcome to go and explore the Flywire, FAFB, and other connectomes in Codex. The URL is on the bottom of the page. I should also mention we do work a lot in collaboration with many of the speakers in this panel, and including Allen Institute, Forrest's group, and the CAVE team, and Neuroglacer contributors. And thank you all for all your contributions.
Thank you so much, Ari. So I was curious from this, is there anything you would have liked to have done in Neuroglancer that maybe wasn't possible, or like a challenge you ran into while making Codex that, ended up being maybe a roadblock with something that Neuroglancer kind of wished it had but didn't?
Yeah, I think it's thanks for the question. I think it's there have been few occasions that we wished, for instance, to have kind of interactions triggered by Neuroglancer so that you can click certain meshes and then respond to those clicks within the application, within Codex. But otherwise, in terms of flexibility and configuration of abilities, I think it's pretty full featured. I don't think we had any limitations there. I think in general, also people like to have kind of like levels of complexity in the interfaces, and I think Neuroglacer would be probably on the more kind of busy interface. I can imagine someone, if they have the energy to do it, to have like a lightweight Neuroglancer version where you can have something that can load quickly, even with a lot of data, and render nicely on any screen size. I think that's more on the UI side.
Yeah, makes sense. Thanks so much, Aaron. Okay, we will move on to the next speaker, and that will be Lars.
All right. Yeah, so I'm Lars Yesterby. I'll be talking about NeuroTrail, which is the neuron tracing and active learning environment. And hopefully that's come through okay. So this combines deep learning algorithms with web-based interface based on Neuroglancer for enabling semi-automated segmentations of brain regions and neurons with a primary use case in light microscopy. So to motivate the need for a technology like NeuroTrail, so neurological disorders affect over 1 billion people worldwide, ranging from Alzheimer's and autism to Parkinson's and PTSD. Drug development is challenging due to incomplete understanding of human brain function. So we've been working as part of the NIH Brain Initiative, seeking better models to understand function and connectivity. And AI algorithms can help tackle these challenges, but there are some gaps, namely insufficient labeled data for AI algorithms, as well as scalability challenges with current annotation tools, especially scaling up for whole human brain scale. NeuroTrail tries to fill some of these gaps with solutions in a few areas. One is an expert in the loop semi-automated labeling capability. Another is leveraging unlabeled data to support unsupervised deep learning paradigms, and then parallel algorithms for large-scale analysis. And so this semi-automated pipeline is essentially trying to improve treatments for brain disorders. The project team that has come together to develop NeuroTrail pulls from both MIT Lincoln Laboratory and MIT campus. The Lincoln team has a lot of expertise in image processing, AI ML, software development, and high-performance computing, which is complemented by the laboratory of Professor Chung at MIT, which specializes in light sheet microscopy, chemical engineering, and neuroscience. The NeuroTrail pipeline that has resulted from this collaboration, as I mentioned, is to enable active learning for efficient generation of annotated brain data for AI algorithms. Walking through this kind of left to right, so NeuroTrail ingests raw data and applies AI algorithms using image processing and deep learning for both neuron tracing and segmentation, as well as brain region segmentation. And then this data is organized by a data aggregation point, which then serves the data into a web interface layer that is based on Neuroglancer. And so the raw data and algorithm results are served to the user. This is built on Neuroglancer. And with the expert in the loop review, users can interact with the annotations, make edits and corrections and updates, and really analyze what's going on with the imagery and the annotations, and then save these back to the data aggregation point, which then is used for retraining the AI algorithms for iterative improvement. And the cycle can continue to support large-scale analyses. I'll first dive in a little deeper on what's going on in the computation layer. So briefly, this computation layer can support multiple algorithms for depending on different use cases and fields of view. Nuclei detection, axon tracing, and region segmentation are three examples. On the axon tracing side, we've developed deep learning algorithms to extract the center lines and trace axon fibers in dense imagery. So an example on the left shows the imagery and then the outputs of a 3D unit. And this is evaluated using the DICE segmentation score, as well as qualitatively looking at where there's true positive agreement with manual gold tracing truth. So in addition to maintaining high accuracy, obviously the one main advantage of automated tracing is the orders of magnitude speed up in tracing time and throughput, which is a big advantage for large scale analysis. So complementing the computation layer is the data aggregation and interface layer, which organizes the data and serves to end users for interacting with the data. And the goal here has been to have a containerized solution to enable scale up for external platforms to really enable the community use. And these tools are also all open source. So as far as data management, some of the schemas we've developed for NeuroTrail include annotation volume chunking, which allows subsets to be loaded and triaged rather than needing the entire data set. Also, diffing, the concept of diffing limits the JSON payloads to only include changes, which obviously is much more efficient. And then having unique assignments per annotation, which allows simultaneous per annotation updates. So on the right, for an example, we can see what the user and client and server would be passing back and forth. If with NeuroTrail's schema, multiple users can update different parts of the data and pass those back and forth between client and server without having to pass the entire data set. And then different regions of the data set can be annotated simultaneously. And so the goal was to facilitate the multi-user collaboration here. And then on the annotation rendering side, NeuroTrail does one-pass high-performance rendering for dynamic length annotation geometry to align with the Neuroglancer baseline. And then there's also per annotation coloring and styling, which makes use of the shader API to highlight specific annotations in different colors. NeuroTrail also has an annotation status flag, which tracks the review status of algorithm-generated segmentations and whether those are corrected or need further correction or review. So the goal has been to enable merge back of these NeuroTrail annotation features to Neuroglancer. So this is a quick demo just to show a few capabilities of NeuroTrail. We have some, so the tool displays large-scale microscopy images of neurons in 3D, and then the AI algorithms generate segmentations of the neurons in the tissue, which users can then edit, manipulate, and correct and search for patterns in the data. In the 3D view, users can also highlight and follow individual annotations of interest like axon fibers. And these can also be filtered by size so that users can focus, for example, just on the longest fibers for analysis. And so the ability to edit these auto-generated segmentations within NeuroTrail significantly reduces the manual data labeling burden and allows for much more efficient data processing at scale. So some impacts of NeuroTrail have been highlighted in a science publication jointly published with MIT Chung Lab. And this is really showing how end-to-end some neuroscience questions can be answered using an integrated pipeline for both the imaging and tissue processing, as well as the data analysis. So NeuroTrail was used for segmenting brain regions, which then enabled counting, doing cell counts in both healthy and Alzheimer's brains. And NeuroTrail was also used for tracing axon fibers in a 3 terabyte dense cortical tissue volume. So just to sum up, yeah, NeuroTrail's goal has been to enable efficient iterative algorithm development and expert in the loop review. The code base is available to the community via open source licenses, and some of these impacts have been highlighted. In future work, I think there's a lot to do and to scale up for whole human brain mapping. So we're excited for that challenge going forward. And here are some helpful links, and happy to take questions. Thank you.
Great, thanks, Lars. Yeah, feel free to post any questions in the chat, everyone. In the meantime, I will fire away. Oh, we actually have one from Bill Katz. Did NeuroTrail let users move through the segmented neuron path?
Yes, right. So in that 3D view example I was showing, highlighting a fiber of interest, yes, the user can follow that through the 3D volume and remain focused on that annotation of interest.
Thanks. I was curious about the process of the review. So it seems like you can edit different parts of the volume at the same time. So does someone kind of check in that they are working on a part and they kind of take ownership for a bit so that there's no conflicts within a chunk? Or how is that handled?
So I think that part is still open to discussion, and I think testing. Certainly, I think the idea is that users would coordinate like a specific annotation region or region of the data set that they would be working on, so that there's no conflicts within a specific annotation. But the idea was to allow the same chunk could be worked on, just different annotations would be updated by different people.
Understood. Thanks, Lars. Okay, brilliant. We will move on to Yael. I can't hear you yet, Yael, but I can see that you're starting to share.
Sorry, I forgot to unmute myself before
No problem.
sharing. Okay. Yes, I'm going to present a little library that we built for our project called the Center for Large Scale Imaging of Neural Circuits. And so, yeah, we haven't changed anything to Neuroglancer. It's really just an entry point to more easily build and interact with Neuroglancer scenes from the Python side. And so for a bit of background in our project, we acquire lots of different types of images in the same human brains ex vivo. So we acquire X-ray phase contrast tomography, which gives us both overview scans of the entire organ at about 20 micron resolution, as well as high resolution images acquired either in excised blocks or in regions of interest in an excision-free manner. And that can get us down to sub-micron voxel size. We also work with polarization-sensitive optical coherence tomography, which is a label-free optical technique that generates this orientation map of the white matter at 20 to 50 micron resolution. And then more classically, we also acquire light shield microscopy and diffusion-weighted MRI. But we don't acquire that data just for their own sake, but also so that they can be used to either supervise or help analyze other types of human data. And so we also need to interact with standard neuroimaging spaces. And so the most famous one is MNI. And it's really just a template, so a mean brain image along with a coordinate space. But there's really many standard spaces in neuroimaging. And so, for example, there's this more recent one called Oxford multimodal, which makes more advanced use of diffusion-weighted MRI. And of course, if we like down the line, we also want to be able to use data extracted from the high-resolution ex vivo data and essentially be able to warp it to single subject space to supervise analysis or inform it in other ways. But that gives us lots of transforms to estimate and to apply, and do that in mostly a dynamic way. Like most in neuroimaging, most often we want to look at the same data in different spaces. We want to be able to move between different standard spaces. So it's not a set in stone thing. Like we want to be able to move back and forth between spaces. And just for a bit of background, the way coordinate spaces are defined in human neuroimaging is really rooted in the way MRI are acquired. So we have someone lying in the scanner, and then we would set a field of view in which data is acquired. And so that comes with a acquisition coordinate space, as well as an image coordinate space, which are usually aligned, but not always. And that link is vendor and sequence dependent. But we also have the fact that the person lies in the scanner means that we can also link the scanner coordinate system to anatomy, right? Because our subjects always lie in the scanner in the same position. And so we know, for example, when you get data off the scanner in a DICOM format, that the x-axis is going to point towards the left of the brain, the y-axis is going to point to the posterior part of the brain, and then the z-axis is going to point to the superior part of the brain. So it's called an LPS coordinate frame. But we can also work with other conventions, and most analysis software actually work in this RAS frame when X points to right and Y points to the entire part of the brain. But it's a nice abstraction, right? Because we don't really care how the data is set up in file. All we care about is having this mapping between voxel indices and this scanner or world coordinate frame. And so when we want to align two images, for example, data acquired in the subject to a template, well, we don't work really between voxel spaces, but we find a mapping between the two world spaces associated with each of these images. So all of our transformations really map between embedding space and not between image space. And that means that if we have multiple images that don't have the same voxel layout or are not tilted the same way, we can still apply the same transform to all of them as long as we know that they live in the same world space. Currently in OMISA and in Neuroglancer, there's nothing that relates to those anatomical spaces, and it makes sense because the aim is to be more flexible and more global than that. But we need those information. So it's in the work to have more advanced orientation metadata in OMISA, but in the meantime, we work with a more simple version where we just store this affine transform in an OMISA data set. And in Neuroglancer, like same idea, it's made to be more flexible than that, so it doesn't know what really anatomical axes are. And that's a bit counterintuitive for people who come from the neuroimaging side, because if they open their data in a neuroimaging visualization software, it's going to be shown the right way. And for us, it means that the software knows that the left is on the left, the right is on the right, the top is on the top, etc. If I open the exact same file in Neuroglancer, it's supported, it opens up, it's shown. But like someone who comes from the neuroimaging side would think that the metadata is incorrect because the brain is upside down. And so that's why we build this tiny library. And yeah, it's really just an entry point. So it's something that creates the correct scene so that things are shown the way we expect them to be. So it's really a bunch of instructions that says, load this image, load this image. I know that this one is a segmentation. And then maybe apply a transformation file and apply a given shader. And so that like now we have a user experience that's a bit closer to what we're used to. And so of course that data is quite small, so we don't need to use Neuroglancer. But we also want that to work with larger data sets. yeah, so with that library, we have 3 different ways of building scenes. We have a little command line interface, which gives us a sort of shell console to interact with a local Neuroglancer instance. We can do the exact same things with a pure Python interface. And then we also have another version that doesn't run a local Neuroglancer and really just creates a JSON state, and then that can be fed to Neuroglancer demo, for example, and just visualized online. And I have another example that is the type of data that I showed before. So it's a bit of a more complex scenes. We have this 20 micron resolution whole brain CT scan. We have different views acquired in different parts of the brain. We also have an MR that has been registered to the X-ray. And then we have a tractogram that was obtained from the diffusion. And so that's all loaded up and set up correctly with just a couple of lines in the interface. And then that's just a classic neuroimaging interaction where we have all different layers, and so people who are used to Neuroglancer just do that stuff. But what I'll show later is like we also can keep interacting with that scene. And so, for example, what we like to do is to look when you have all of these layers acquired with different orientation. We might want to be able to we might want to look at the data in a frame that is aligned with the voxel space of a given layer. And so that's something that we can do easily by with our library, and go back and forth between those views. Yeah. So I just acknowledge the collaborators who are part of this project, and happy to answer any questions.
That's great. Thank you so much. I'm actually really curious about what drew you to Neuroglancer. I know you mentioned part of it is like that it can deal with larger data. Part of it I've seen there, maybe the integration with Python, but yeah, from your perspective, what drew you to, or drew your team to Neuroglancer as opposed to yeah.
Yeah, good question. So it's really the size of the data, right? So we used to, like, I think one of the labs comes from the MRI side, and so by moving to ex vivo imaging, like we work with larger and larger images, but still not to a size that needs something like Neuroglancer. But now we are acquiring those X-ray data sets that can't be loaded in memory at once. So I think one of those scans is probably a terabyte in size, or up to 10 terabytes in size, depending on the field of view. And then our large data is also becoming bigger and bigger. So we had to start working with something that can deal with the larger data sets. But and then the Python interface made it easier to kind of build these little tools to make it easier to work with the metadata that we use to from the analysis point of view.
Perfect. Thanks so much. Okay, we'll move on to the next speaker, who is Stuart.
There we go. Hi, yeah. I'm the project scientist for FlyEM, as I mentioned. We are in the business of making connectomes. We're close to releasing a Fly whole CNS connectome. We have a couple of other connectomic projects, notably this zebrafish volume in collaboration with some folks at Harvard here, you see. I thought I would, so our primary workhorse proofreading platforms, Forrest already gave a nice explanation of what proofreading means in connectomics. Our primary workhorse applications are desktop applications, but Neuroglancer is like an indispensable part of our workflow, and we're trying to, increasingly use that and eventually dispense with our desktop applications. But for now, we implemented a couple of tools that incorporate Neuroglancer for doing a decent subset of the work that we do. So I'll talk about 2 of them. The first is a platform we call Clio, and I think I'm going to breeze through a couple of the features on the slides here, and then if I have time, give a quick demo. But the basic idea, it's a web app. It incorporates Neuroglancer. I should mention that the front end is primarily the work of Jody Clemens and Philip Hubbard here, and the back end is thanks to Bill Katz. It incorporates Neuroglancer as a component, and a couple of speakers earlier have already mentioned how that could be a challenge. I'll say a few words about that a little bit later. It offers users the ability to drop their own annotations, which get stored on a server. An important part of putting together a connectome is actually annotating all the neurons by cell type and so on, as Arya described. So we offer a feature, a system here for people to query for neurons that they're interested in, pull the data out of the database. The database is all in memory. It responds pretty quickly, and then they can actually edit the metadata as they see fit, send those back to the server. As I mentioned, our primary proofreading tools are not based on Neuroglancer, but Clio does have some lightweight proofreading tools, including the ability to submit your own merges to the data set. Now, these don't get applied live, but rather kind of you perform merges, they get applied in your own view here, and then you submit what we call a pull request to be processed offline, and as a way of contributing to the data set if you don't want to install our desktop applications. In addition to that, though, we've implemented what we call proofreading protocols, which are a little bit more streamlined way of doing, I guess, for lack of a better word, assembly line style proofreading, where you can queue up large batches of tasks, sometimes very focused tasks. So this is a screenshot of what we call focus proofreading, where maybe we have some algorithm that has come up with additional merges to propose, but it wasn't confident enough to include in the original algorithm, in the original agglomeration. So the proofreader is presented with a decision. Okay, should the green thing and the yellow thing be merged together? Yes or no? They can just answer yes or no, or maybe there's some edge cases. We offer them up here, and then churn through thousands of these, well, hundreds to 1,000, let's say, in a day. There's kind of a related or similar idea here, but here we're not asking if things should be merged together or not, but we're reviewing zebrafish synapses to be used as ground truth for training a model. We can ask if this is good synapse, bad synapse, false prediction. Similar idea. The whole thing is backed by a data store, what we call the DIVID for the, I guess, distributed version image data store, which I won't say much about here, except that I guess the annotation bit is in memory, as I mentioned, and it's able to provide checkpoints for each version of the segmentation and related data sets. It serves up all of our skeletons, our meshes, all the segmentation voxels, and so on. And then the other tool I'll say a little bit about is called Newprint. It's what we use to actually publish the data set. It's based on a Neo4j graph database, again, with a front end implemented by Jody, and it offers various options, ways that you can query the connectome, figure out what's going on. Maybe I'll just show you one recent feature that we added. Sorry, I'll just share a different window here, because I think it's relevant for other Neuroglancer-based apps. So one of the complaints, or let's say challenges from new users, is that, and I think somebody mentioned, right, that Neuroglancer, or I think Arya mentioned that, the Neuroglancer UI can sometimes bewilder people. I love it personally. But we were trying to think about simple ways that we can offer users some relief there. And one is just the ability to just offer presets. So you need, okay, if I don't know how to turn on and off the segmentation, maybe that's kind of obvious. Or if I want to see the tags that are available on this data set, or I don't want to see them, so on. Now, this requires essentially that your outer app is able to poke state into Neuroglancer and pull state out of Neuroglancer, which can be kind of tricky. So I'll get back to my slides here. I'm trying to think if there's anything else I wanted to say here. I think this is okay. Sorry, finding my slides again. So the answer is in a component that Jody built, and you'll have to ask him about the details if you have any questions, called React Neuroglancer. So both of the applications I just showed you are based on React, and then to integrate Neuroglancer into them, Jody wrapped Neuroglancer as a React component, and that is really the trick that allows us to, when things change in Neuroglancer, that state can bubble up into the appropriate callbacks, and then the application can become aware of it, and then it can push state back down into it. Now, in order to get this working, I believe we had to make a fork of Neuroglancer, but it's not. Originally, it wasn't a major divergence, but over time, it's grown to diverge, and so we're in kind of the same situation that Forrest mentioned they were in, years ago, about like having this divergent code base. It would be nice if we could think of some ways to kind of incorporate the necessary features that we had to add to get this to work into the main line. As I understand it, basically kind of the general theme is just that Neuroglancer is like excellent as a standalone app, but that it makes a few assumptions that it is the standalone app. And so then you have to kind of patch it in some ways to get it to cooperate with, as part of a larger system. And so one of those is like, don't change the URL every time the user clicks something in Neuroglancer. You have to have a different build target. You have to be careful about when the right-click menu is actually disabled so that it can function in Neuroglancer, but it still functions normally outside of Neuroglancer. And then things like styling leaking out of the application and so on. There's an example on the GitHub repo if you're interested in trying this in your own web apps. And then the last thing I was going to mention, Forrest also mentioned that they're getting good mileage out of this, and I was just going to flag this for those who haven't tried it. Being able to mock the Neuroglancer pre-computed format is super useful, and it seems pretty well designed actually for that use case. And so we're able to just stick little, when we want to add, okay, annotations, customized annotations, or synapse counts, or different formats for our meshes and things like that, we can just stick them in a little Cloud Run function on Google and then make them available for users to use. Now, this requires, that if the user wants to exploit these, you have to either bake this into the URL that you gave them, or if there are options in these little URLs here, they have to kind of edit it by hand. So it's maybe not the smoothest user experience, but it can be super useful for those who are, hungry enough to make use of it. And then the last thing, this is just like kind of a detail, but we also have a link shortener for Neuroglancer state. I know that there are other link shorteners out there for this for users, but it's worth looking into if you are getting tired of sending around huge Neuroglancer links. Code's on GitHub if you want to deploy it yourself, or you're welcome to use ours. And that's all.
Perfect. Thanks, Jordan. I was wondering, you touched on some of them for sure, but if there was some really key points of what Neuroglancer could do to make it easier for integrators in that community, do you have any ideas there?
I think Jody Clements, who's I think in the audience and maybe could reply on chat, would have a much better idea of what's necessary there. I'm not as much of a front end person. I guess I've heard Forrest has thrown the term around integrator in Neuroglancer terms.
Brilliant. Yeah, and Forrest thoroughly agrees with what you said. So thanks so much, Stuart, for the talk. We'll move on to the next speaker, who is David.
Okay, let me share my screen. Okay, hi everybody. Today I'm going to be talking about CellMap Flow, which is a tool that we've been developing for real-time on-the-fly inference of arbitrarily large data sets using Neuroglancer. So here, this movie is showing CellMap Flow in action. So this is just a Neuroglancer view where the raw data is an EM data set shown in grayscale, and the colorful layers correspond to organelle segmentations. And these are actually distance predictions that the segmentations are, and they've just been thresholded to give an idea of like what the semantic segmentations would look like. And the important thing to note here is that this is all being calculated on the fly. This is not loading the predictions from disk. It's taking the raw data, feeding it through a model on a GPU, and then returning the predictions back to Neuroglancer for viewing. And so the problem that we're trying to address with CellMap Flow is rapidly getting from raw data to segmentations. And the big problem is that there's often many steps that are involved in practice to get from one to the other. So often there's pre-processing of your data, running it through some kind of machine learning model, and post-processing. And usually, if you have to do this like a single time, that's not too bad. But oftentimes you want to test out a variety of pre-processing parameters, maybe test out different models or different iterations of a single model. You might want to test out different post-processing after you get the predictions from the model. And usually, especially if you have like data sets like we do that are many teravoxels in size, you probably want to see how it performs in different regions. And so, as you need to test in different regions, the complexity of keeping track of everything just grows exponentially. So the idea behind CellMap Flow is to act as a way to get this on-demand inference with a suite of customizable and extensible tools. So the idea is that you can, with CellMap Flow, get inference anywhere in a data set, and the data can be both local or hosted on the cloud. We also provide a variety of pre-processing and post-processing algorithms built in to for before you run your model and after you run your model. We also provide a variety of pre-trained models and allow the user to be able to easily run existing publicly available pre-trained models like CellPose or bioimage model zoo models. And it's very easy to change the parameters in these algorithms, and I'll show that in a second. And also easy to extend and add your own custom models or pre-processing or post-processing algorithms. And we also provide the ability to save parameters so that you can process the entire data set after the fact, also using CellMap Flow once you decide what worked best. I'm not going to go too much into how this works since the time is short, but this is a slide I'll show to kind of describe it. So the idea is that we wrap a local Neuroglancer instance in a dashboard, which is shown here. And the dashboard has the ability to select different models that you might want to run, the different input algorithms and the settings for those, and also the post-processing algorithms. And when you run CellMap Flow, it spins up one server for the dashboard and the local Neuroglancer instance, and another server to intercept the chunk requests from Neuroglancer and return process data. So this has, I think, been touched upon earlier with kind of we're mocking or setting up a virtual data set where we point Neuroglancer to that data set, and when it makes a chunk request, we take that chunk request and we crop the relevant region from the raw data, and then we feed that through whatever pre-processing steps were selected, feed that through a model that's been loaded onto the GPU, and that model has been decided either when you spin up CellMap Flow to begin with, or you can select it from the menu. And then we take the results, the prediction results, and send that through the post-processing steps, and then feed that back to Neuroglancer as the return for the chunk request, and then it loads it in the viewer. And so CellMap Flow automatically takes care of, if you have multiple models running, it'll spin up another server for each model. So here, for example, I'm showing two different models that have been chosen, and each one actually has 6 output channels for the prediction. I'm only showing one of each, blue corresponding to mito from one of them, and red corresponding to mitomembrane for another one. And as you browse around the data, as we showed earlier, the predictions are actually being calculated on the fly. And so this is obviously just a snapshot, but to give an idea of the speed that we get. So sorry, this is a command that you would use to run CellMap Flow. So you just tell it the path to your model, the path to your raw data. And this model is actually outputting 8 different channels. And again, this is a distance prediction that's been thresholded for semantic segmentations. And you can see how responsive it is. So for every time a chunk loads, it's processed the raw data through a model on the GPU and returned the results to the front end. And so you can easily browse around huge data sets and get very interactive results. And for the last couple of minutes, I'm just going to walk through sort of a more concrete example that shows a few more of the features. So in this case, this is how we would start up CellMap Flow. You basically only have to point it to the raw data. And in CellMap Flow, if you have local models stored somewhere, you can, in a YAML file, point to those, and then it'll automatically populate the menu and the dashboard with those models. So that's what's going to happen, in this slide. So the user is going in and they're going to go to the models. They're going to select the relevant model and click submit. That'll spin up the server for the model and start doing the inference on the fly. And so here, this is mitochondria predictions. And again, it's distance. So brighter pixels correspond to pixels that are more likely to be part of a mitochondria. And then the next step that you can do is you can, oops, sorry, it's the same slide. So here the user is going to add a threshold post-processor, which basically converts the distance predictions to semantic segmentation, so actual segmentation layer, where the blue corresponds to mito and the rest is background. Then next we add a blockwise connected component post-processor. So within a block, we take this semantic segmentation and do connected components. And so you can see now each individual connected component within a block is given a unique ID. We also provide the ability to merge across blocks. So any IDs that are touching at the boundaries between blocks will be merged, and this is updated periodically. So you'll see first it'll load chunkwise, and then it'll load, then it will merge touching items. And so here you see, for example, this is like 4 mitos that are considered part of the same object, but they actually shouldn't be. You just have these sporadic pixels joining them. And so the user is going to update the threshold parameter to raise it to make the threshold more restrictive. So that successfully splits up the mitos. And this has really sped up our processing time, because now we can explore entire data sets and test out all of these different parameters to see what works best, compare different models or different iterations. It's really taken us kind of from the order of months to the order of minutes or hours. And with that, I just want to thank everyone involved, especially Marwan Zwinky, who put in a lot of effort to get CellMapFlow to where it is, Jeff Rhodes, and also more recently, Larissa and Yuri, who have been contributing to CellMapFlow. And this is the GitHub repo for it. And as I pause to take questions, I figured I'd just show a few more examples of what it can do. So we have the ability to do input normalization. So this is like a pre-processing step. We also can process arbitrary types of ML algorithms. So this is doing affinities for neuron segmentation. And you'll see the neuron segmentations pop up as the movie plays. This is running it on cloud data, so on like Google Store. This is loading bioimage models on local data. And this is running cell pose models on light data. And that's it.
Great, thanks, David. Yeah, it's a really interesting use case. I wonder, do you think, that there's maybe an untapped potential to some extent for Neuroglancer to act in this way more often, for people to see these kind of dynamic visualizations of what they're doing with models or other things? Yeah.
Yeah, I think I never even really considered this. Stuart actually showed me an example he made just to kind of spoof a N5 or ZAR, where it would just show different colors, and it could be arbitrarily large because it's just serving up the data as you browse around. So that's what kind of inspired this, that you can get this kind of more live feedback, because the Neuroglancer examples that they have for Python show kind of live inference, but you have to like click a button and then it will take the region around that to process. But this way turns out to be very useful, because you can just browse around and and have it do it for you. So.
Yeah, absolutely. Yeah. It reminds me of like when I first started using Napari, for example. Those kind of examples are maybe more commonly seen in Napari, right? In Neuroglancer, maybe they're not as regularly seen.
Right.
So something like this, right, really highlights the power of kind of that thing you can do.
Right.
Great. Thanks, David.
Yep.
Okay. And up next, we have Matteo. Will be our last speaker.
The last speaker is there.
Last but not least.
Some people are still connected, so that's good. Thank you, Sean, and thanks all the speakers. This has been great. Is my screen showing? Yes.
All good.
We don't know what Neuroglancer is, but yeah.
You're not in full slide mode is the only thing.
Yeah, that's fine, because I'll be switching.
Cool. Just make
it. Should be faster. So as far as was mentioning at the beginning, is MetaCell will be contributing to some of the features on Neuroglancer, sponsored by Allens, by CZI, and so from our perspective, we have seen from the beginning, how a great engine was, and here are some of the things that make it so. Obviously, the fact is, you guys mentioned earlier that you can visualize, large scale 3D data set on the browser. It's kind of the fastest engine out there, which is, I think, one of the reasons why many people have been using it and retrieving chunks of dynamic size based on the viewport. And volumetric rendering, Forrest earlier was showing the latest that we could also contribute to. And like one of the things that I think made Neuroglancer great is essentially the fact that the entire application state is encoded in JSON, and that is in turn part of the URL. And that makes it easy to collaborate, because essentially you can, it enabled something that was not trivial for large volumetric data set. Like you have a link, you give it to someone, and that someone, no matter where they are in the world, they can look at the same thing that you were looking at. And this has been great. On the other side, this also started posing some challenges from what we've seen. Like these URLs start becoming larger and larger, for instance, and this means that tools have troubles at times to even share them. We've seen people using Google Docs, for instance, with the collection of different Neuroglancer links. Also, we've seen how all links, in a sense, look the same, but they do not necessarily represent the same concept. There are some links out there that are meant to just be snapshots. Okay, this is this particular view that I want to share, and I want it to be an immutable. Other links instead are more like stable data sets, so links that essentially people will be working on and that will evolve over time. Links that essentially people will want to collaborate. And we've seen how there's different tools out there that have been trying to, tackle and work around some of these things. So keeping track of all of these links being daunting. Tracking what we've learned is not easy, because there is essentially, by design, there's no backend, which is one great thing. On the other hand, it also poses some limitations. The moment that, for instance, you want to find and track some of the things that you've been working on, we've seen how third-party software is usually involved to tick the boxes on some of those scenarios. There's no way to track the changes that I made to a visualization and to share with specific people and know what I'm sharing with different people. The interface is great on one hand, but it's also an expert interface. So for newcomers, it can be a bit daunting. And as we've seen, different custom applications are usually required to satisfy different workflows. So today, I will present Neuroglass, which is something that, as MetaCell, we've been building in the past year. It's still currently in development, and it is a web application to facilitate storing, sharing, and organize Neuroglancer-powered visualizations. So our goal is to help scientists organize and find their data, removing the need to create grassroots solutions to handle Neuroglancer links. Essentially, the idea is to take anything that is not workflow specific, that is not domain specific, and just cuts across anybody that is using Neuroglancer, and make that functionality available for everyone. Give a way to easily share and collaborate on Neuroglancer states, and provide a unified user experience, as I was saying, when managing Neuroglancer workflows of different kinds. Reduce time that is required to create also ready to publish images and artifacts. I will show some about this in the next minutes, and ultimately also not remove, but rather reduce Neuroglancer onboarding, make it faster for new people to learn the ropes and see how you can go about doing different things. So here I have in a few slides some of the functionalities that we have, and then I will do something less wise than what you guys have done, which is do a live demo. I should have recorded it in hindsight, because I know that things will break, but nonetheless, I like the thrill. So we will see things as short URLs that is essentially available in order to collaborate, and having something that can just be easily sent to anybody. We see that the tool has functionality to share the studies themselves to have multiple editors working at the same time on the same Neuroglancer state, and the ability to have extensive search. I won't spend too much time here on the static slides, such that I have more time for the demo later on. We will see that we introduced essentially two concepts to cater to these different concepts associated to the links, as I was saying earlier. One concept is studies, and one concept is glances. And we will look at that in a second. History is also something that is part of Neuroglass, so anything that you do is tracked, and you can think of from this perspective, like Google Docs for Neuroglancer is often the way we think about this. We have the ability, building on top of what Forrest was showing earlier, to have figures and having figures that can always go back to reproducible states that then can be forked and so on. Tagging to organize the different studies, and of course, metadata. Let me hop onto a quick demo. So this is what Neuroglass looks like. So here you have the dashboard where you can find studies being those places where you keep doing the work and you keep updating them. Each study is, we will see, so we'll just go inside one of these. I think a lot of what I will show is coming from the album in terms of data. And let me minimize Zoom, that is hiding some of the interface. So here you have the Neuroglancer that everybody's familiar with. We polished the look and feel and the styling. So you will see things here on the right-hand side that kind of look a bit more polished compared to what you have usually seen. We have the history, which is essentially any changes that you do. You see there is a save icon, and therefore you can go back to any modifications that you've made. There is the, as I was mentioning earlier, the ability to share a study with different people, usual permissions that you can think of finding in Google Docs, like applications. There are figures, which are snapshots for a visualization. So this is a particular figure. And one of the things that you can do here is to always go back to the glance that was used in order to generate that particular figure. And this is something that it is now loading. I hope. I know I have little time. I want to show you some things that you can do here. For instance, you can search for any keyword that you may have in mind, and this will essentially search throughout all of your studies and glances. And so this will tell you, for instance, that in this particular study, there is one shader in this layer that is using the envelope function, okay? Very quickly on, this is our dev version. This is not yet released, but we're introducing also the ability to use AI to interact with Neuroglancer states, and this is to try and do things that, at first the people may not know how to manipulate the shaders and so on. So this is a prompt that I have here that I'm submitting, only shows synapses with, for instance, a score above 800, do not show autopsies, and color the whole annotation as purple. So this is now processing the data, and this outlines the changes that were made to the state, and modify the synapses layer to only show, etc., etc. You can expand this, and you can see exactly what changes were made to the Neuroglancer state, and you can iterate on this as you are used to do with other similar agent-based AI tools. Data collections are a way to bring in multiple data sets at once. I'm skipping a bit because I think this is about how much time that I have, but you can essentially by ingesting a CSV file, bringing in multiple data sets, each of these can have multiple layers. And then you can retain the same visualization, which we're calling lenses. So you can retain the same lens that then can be used in order to look at different data sets in the same way. So now, for instance, I just switched from data set 1 to 5 using the same lens. And I will go from data set 5 to 9. It's using the same lens. And this is different, essentially, a set of data sources that are being used. I stop here because I think I'm out of time, although I'm not being correct.
Yeah, bet on time. Yeah, thanks, Matteo.
Okay, sorry if I went 1 minute long.
No problem. Yeah, curious how you see the integration with other tools and resources maybe happening for Neuroglass, and possibly even some of the ones we've seen today. Could you see Neuroglass maybe using some of those in the future?
Well, absolutely. So one of the things that we want to give as the possibility, first of all, is for every group that is interested in using Neuroglass to use their own fork as well. Of course, the fork would need to be kind of Neuroglassified such that it can integrate with all the functionalities that we added on top, but that would be a way to kind of not kind of duplicate workflow specific applications for sure. The other thing is in order to help also the community kind of factoring out the things that are needed by everyone, also by doing things like what we're doing today and learning what different applications are doing, and essentially offering our help also to contribute this back to the main Neuroglancer. I didn't mention that we're already starting opening pull requests to the main Neuroglancer repo for some of the things that we have added through Neuroglass in terms of, like, for instance, the user interface. Now we'll be contributing the accordions, for instance, that we're using in our UI, and little by little, as everybody that worked on Neuroglancer knows, it takes a bit of time, especially to have a good PR that Jeremy would be happy to merge, but we started kind of doing this, and we'll keep doing so.
Great, thanks, Matteo. Okay, I just want to point out as well, similar to what Matteo mentioned here, there's been some really great chat that happened from everyone. So thank you to all the attendees and all the speakers who really participated. Some very interesting stuff. I can see threads of how people are thinking, how these could be combined together. What are some common pain points people have? How could this go in the future? So thank you for the people who listened to this and contributed those ideas. I think we have time just for probably one key takeaway left, very quick from each speaker, and then we'll wrap up there. So we'll go in speaker order. Forrest, key takeaway.
Well, I'm just really impressed with the variety of speakers and how many different things people were able to do building off of a common platform. And I think it's a testament to the design of how Neuroglancer made it possible for people to go in and plug in different things that are doing different features at different levels of integration. So it's really an impressive community who's doing a lot of really cool things. So thanks, everyone.
Yeah, absolutely. Yeah. Ari?
Yeah, repeating what Forrest said, it's really evidence for the initial design principle for Neuroglancer. It is scalable and flexible, and hope to see more in the future, more advances and more improvements.
Great. And Lars, any key takeaway or something you want to add to?
Yeah, I echo that, and I'm excited how these technologies can work together to continue tackling challenges in the neuroscience community. So yeah, really excited to be a part of it.
Great. Yeah.
Yeah, I'm super impressed by everything that was presented. And I'm really looking forward to this kind of federated way of like taking little services that serve pre-computed data but do smart stuff behind the scenes in order to extend it like without having to put everything back into the main line.
Yeah, absolutely. Yeah, it's been a lot of interesting applications. Yeah. Thanks, Stuart.
I think I'll just echo what everybody else is saying. It's great to see very impressive talks, very exciting future, I would say.
Brilliant, yeah. And David?
Yeah, it's been great to see everyone's projects and just to be aware of what's going on, both for collaboration and also to avoid duplicating ideas and see what other people have developed.
Yeah, totally agree. Matteo?
Yeah, I'll go last just by saying, yeah, thank you to everyone. I didn't mention because I was running out of time. I also wanted to acknowledge the precious input that we got from different folks like Forrest, David, and many others. So we've been showing what we were doing, and they were happy to give us feedback and steer us in the right direction. For instance, the AI thing I was showing, it was actually, I think, Forrest direction in terms of like, this is a way that AI could be useful. And I think it's by a kind of keeping together the community and keeping open this dialogue, knowing what everybody's doing, that you know, we can keep making progress. So thank you all for attending, and thank you, Sean.
Yeah, thanks so much. That was really, really great. Fantastic discussion with everyone and the speakers. So thank you for coming to the webinar. Hope you enjoyed, and see you at the next one.
Thanks, everyone. Bye-bye.
Cheers.